Thank you for attending today's webinar, Why So Pale and Wan? Comparative Genomics of the New Cavefish in Europe, presented by Dr. Fritz. Dr. Sedlazeck completed his Ph.D. in 2012 in the group of Dr. Arndt von Hassler at the Max F. Perutz Laboratory in Vienna. After a 2-year postdoc, he transitioned to the lab of Dr. Michael Schatz at Cold Spring Harbor Laboratory and later to Johns Hopkins University. Since 2017, he has been leading his own group at the Human Genome Sequencing Center at Baylor College of Medicine. His research focuses on developing computational methods that detect and analyze genomic variations with a focus on structural variations. He and his group are working on very fine-scaled applications to detect variations that can explain certain disease phenotypes, as well as large population-scale applications to deepen our knowledge of structural variations. Without further ado, here's Dr. Fritz.
Yeah, thank you so much, and thank you to Genomics for giving me the opportunity to present my work. My name is Fritz. As mentioned, I'm an assistant professor at the Human Genome Sequencing Center. There, my group is mainly focusing on structural variants and also phasing of different variants to detangle and better understand diseases in humans. However, today I'm not going to talk about any of this. I'd rather focus on this very interesting little, not so appealing cavefish that we are currently studying in collaboration with the University of Oldenburg, as well as with the University of Constance, both in Germany. So you might ask why I'm switching from humans to cavefish, and that has some multiple reasons that I'm going to convince you about over the next minutes. So why do people in general studying cavefish is actually quite interesting. So as you know, these cave animals or cave fishes, in this specific reason, are really animals that inherited environments that are quite different to our normal environments, meaning they are of course dark, less foods, and often also cold. So however, evolutionary wise, it's quite interesting to study these animals because they have adapted very rapidly, or they have adapted over millions of years to these environments. So first of all, they over mutations, they had lost their eyes or lost sight because it's not necessary actually to see in the dark caves, versus for us eyes and sight is very important. But therefore they increased their ability to censor their environment and censor themselves to not swim against the cave walls, for example, or other stones or other fishes. They also had to change quite heavily their metabolism because, as you can imagine, these caves, it's quite a harsh environment and there's not much food to be found in general. And also they most often, or almost always, lost actually their pigmentations, because again, if you cannot see someone, why would you pay the extra price to camouflage yourself, right? So it's been quite interesting and fascinating to kind of dive into this project. And actually I want to point out this very interesting homepage or page, which is cavefishes.org.uk, which has a nice collection of all these different cave fish species that are currently around and known. We are still finding new ones, such as the one that I'm presenting today. But in overall, this homepage holds already 326 different cavefish species, so quite much more than I ever thought are going to exist. So what makes our cavefish that interesting makes me switch topics from human genetics and human disease genetics to this little fella. It's quite straightforward. So in around 2015, Joachim, who is a hobby cave diver, and his friends investigated this cave near Aach in South Germany. And what he found over the 4 to 6 hour dive in this cave was a little fish on the ground, almost like 15 centimeters, very small, that had apparently adapted this environment. So he brought one of these species up in 2015 and showed it to Jasminka, one of our collaborators, and she immediately recognized the importance of these finds. So over 2016 and 17, they collected multiple samples of multiple fishes around these caves, indicated here by these different dots that are yellow and red, marked yellow and red. And it's really astonishing. So again, I just want to stress this point that this is kind of really labor intense. So in the first part of the cave is accessible from outside, still like a 4 hours dive to get through this cave. But then the later part of this cave, since the middle part is collapsed, you really have to go through this cave system marked here in orange, and then do another around 4 to 6 hour dive to collect those samples. So it's really thanks to Joachim, that we could collect all the species. Of course, we didn't want to oversample this population so that this population is still livable, but we wanted to collect some samples which had happened over the last 3 to 4 years. So what makes our cavefish so interesting and special? Well, on the left side, you see I put here pictures from the surface and the cave population of the Mexican cavefish, a very well known and very well studied fish species that led to multiple nature publications and so forth, and really great insights in the development of these cavefishes in this particular species. However, this cavefish splitted from its surface population around 4.6 to 1.1, 1.2 million years ago, so quite distant. And over these millions of years, you can see it developed. He kind of lost his prickling or lost his shiny surface. It also grows skin above his eyes, so he doesn't have any eyes anymore. I hope you can see that very nicely in these two pictures. And also had other adaptations. Our cavefish, on the other hand, as you can see on the top is again the surface population, and on the bottom is the cave population, has also developed some of these traits already, maybe not as strong as this Mexican cavefish, so you can still see a reassembling of the eyes, this little black dot in the cave population. The interesting part here is that it's estimated to be around 15 to 12,000 years ago since those two split. And one of the reasons why we estimate this age is that the cave itself was dated to be maximum 20,000 years ago, when the glaciers were actually still around this region in Germany. So since the cave is not much older, it cannot be that the fish is that old. And also some microarray analysis also confirmed or indicated this age. So it's quite remarkable that within this few thousand years, which is actually just a split second in terms of evolution, these traits and this adaptation already took place in this cavefish. So just to kind of bring this point a little bit more home and illustrate it a little bit more, around 400,000 years ago to 40,000 years ago, that's around the time that we are estimating the Neanderthals to be around. So they got extinct 40,000 years ago. Just a random fact, around 13,000 years ago, the giant sloth got extinct. Quite a nice fella. What is more interesting, 13,000 years ago, Aleppo, the city, was founded. So this is the oldest still populated city of humans. So around the time when this cavefish split from the surface population was around the time humans got civilized, settled in a city, and still are living in this city. So that's kind of a perspective how recent this actually is. And then around 12,000 years ago, maybe a little bit before that, our cavefish split, as I said. Other interesting facts are Jericho, one of the other oldest cities that are still populated by humans, was founded around 11,000 years ago. 10 to 12,000 years ago was around the time where we domesticated goats and cattle. So you see really all around this time the human civilization took place in the domestication of our environment. And just to give another comparison point, the pyramids, the oldest pyramids, are currently estimated to be around 6,000 to 7,000 years ago. So just over this very recent time scale, as you can see, the cavefish develop mutations that lead to reduced prickling and camouflaging, as you can clearly see in these two pictures. The eye size has been reduced significantly from here to the cavefish population. And then also the sensory unit. So you see those little tentacles in the beginning, or on the snoot of the surface population, has been enlarged in the cave population to help it to orientate him and be safely swimming in this case. So this is very interesting from an evolutionary point of view. As I said, these mutations have been really recently. So we kind of sat down and thought about, okay, what do we really need to study these mutations and study these strong selections and which genes are really affected here? Well, luckily we got lucky in 2018, and we're the winner of the PacBio Smart Grant, sponsored by PacBio and from GeneVerse. And this really kind of spearheaded all this kind of analysis that I'm going to show you and is also still in progress, which kind of made me from this little guy sitting in the back to this little guy jumping through money and genetic data. So with that price, or with that money, with that sequencing capacity that we obtained, we wanted to study the genome, because there is no reference genome yet for any of these loaches, so no close relative reference genome is available. Then we wanted to annotate this genome, of course, and the most interesting part here is to also obtain transcriptome annotation, not just gene annotation, which should further improve our understanding of the orthology between these cave fishes, so between our cavefish and the Mexican cavefish and other cavefishes where data is available, for it's sparse in other cavefishes. And then we wanted to, of course, obtain data sets and insights about the population itself, because it's not enough to just have a genome and have the genome beautiful annotated. You need a sense of how the population is formed and which mutations are common and rare, and which of the two populations, being it the surface or the cave population. And for that we have done Illumina sequencing to obtain allele frequencies, FSTs, and other common measurements to better understand the evolutionary process and the genomics and genetics of these populations. So for genome sequencing, we sequenced the genome together with GeneVis. Here is one of those plots that I like the most to study the quality of the DNA reads that we obtained. So on the left you can see the coverage. The genome of this loach is estimated to be around 600 megabases to 700 megabases. And on the x-axis, you can see the minimum length cutoff of these reads. So we're starting without a length cutoff on the left side, and then applying different length cutoffs as we go along the x-axis. So you can see in total, for this around 600 to 700 MB genome, we obtained a coverage of 80 plus X, which is very sufficient for diploid organism to be assembled very nicely, especially of this size. And then what this plot is telling you, that if we set up a threshold of around 18 to 20 kb long reads on the x-axis, we'll still have a coverage of around 40x, which again fosters to obtain a high quality contig assembly. So with those reads obtained, what we did, we were interested in how different assemblies were forming here. So we used Falcon, very well known and established assembler specialized for PacBio data. We used WDTBG2, which is a more recent assembly, especially quite powerful and very fast to run. And we used Canoe, which is also a very well established long read assembler capable of assembling PacBio as well as nanopore data. So on this plot that I'm showing you here on the x-axis, you have the different de novo assemblies. On the y-axis, you have the different contact lengths, and you see the viscous plots of these different assemblers. As you can see, Falcon is a little bit of a buff about the length distributions compared to the other assemblies. WDBG2 is in general showing the most diverged contact lengths, which I'm still not sure how to interpret that compared to the others. So, but in general, you can see that Falcon actually produced not just the largest N50 size, but in general the largest contigs out of these data sets. And that's one of the reasons why we chose that assembly with an N50 of almost, not quite, but almost 1 MB, which is quite astonishing. And also the total size of Falcon, but also of the other assemblers, kind of fit our expectations of being around 600 to 700 MB genome size, which, as you might or might not know, these are just very rough estimates from biology. So the other reason why we used Falcon is because it also provides the method Falcon Unzipped as a sub-processing part. So Falcon Unzipped was published in 2016, where I also contributed to this paper together with Jason Chin, who was the lead author, together with PacBio on this paper. So what Falcon Unzipped does is not just providing you one set of contigs, but it also goes back, investigates different bubbles and therefore SNPs and structural variants in the assembly graph itself, and tries to see which of these regions can be phased out. And these phased out regions are then called haplotics. So you can see that here indicated by these red bars. It is important to know that although the primary contick is continuous, we don't know the relationship of the haplotypes between haplotick 1 and haplotick 2 because we lack this information in these gaps. But it's still, I think, one of the first or the first de novo assembly that gives you insights not just on one strand of DNA, but basically on the diploid state of your DNA, what you want to investigate. So running Falcon and SIP with the help of Sarah from BacBio, we obtained haploticks as well. The N50 size of those haploticks are 98 kb. Again, these are just the regions that could be phased. However, what was interesting is that the total size of these phaseable regions is 414 MB, which results in kind of a phasing of almost 70% of the contigs that we have. So this is not just one of the first, or the first, as far as I'm aware, loach genome assemblies that we have, but further on top of it, we have it almost in a for the majority, we have it in a diploid state, as it is actually in nature and biology, and can assess the different haplotypes. So now that we have this genome, we of course wanted to go the next step further and annotate this genome. And for that, again, BacBio as well as GeneVerse was so generous to provide isoseq data for our fish. And with this isoseq data, we want to study which genes are impacted, of course, or which genes are carrying these mutations between the surface and the cave population, but also, of course, narrowing down this gene candidate list or this isoform candidate list, giving the orthologous sequences or the orthologous genes between the different cave fishes, being it the Mexican cavefish or other. So when we look what we obtained is the following. So using isoseq3, the pipeline that developed by BacBio, we obtained 58,600 potential transcripts. As you can see on the left is the histogram of showing you the transcript lengths that we obtained. It's nicely centered around 2 kb, what you would expect as kind of a gene length or isoform length. However, as you can see, we have a very few, but still a couple of potential transcripts that are as long as almost 10 kb, which we have to study if that is kind of a real thing or some sort of an artifact from the pipeline itself. Overall, we are currently in the process of optimizing these parameters a little bit, as well as looking into other methods, which is called TAML. So the next thing is, of course, take this potential transcripts and make some sense out of it. So what I did here is a very straightforward analysis, simply mapping these potential transcripts back to our contigs and collapsing or clustering the overlapping transcripts, and therefore counting just like unique regions. And these unique regions I'm referring to right now as potential genes. There are of course several biases that can happen just because of this strategy, but we wanted to get an idea of how many genes we potentially have captured with this method. And for this kind of fish with this kind of genome, we would have estimated to obtain that this fish has around 16,000 to 20,000 different genes. So when I'm doing this kind of very cruel and straightforward analysis, we are ending up with 16,400 potential genes, which fits kind of the expectations we had. Of course, one driver that this number is a little bit on the lower side is that if regions were not assembled in the genome, I will not be able to map it back, and therefore I'm not able to assess them if there are additional genes in regions that have been currently not assembled as full. So what you can see on this histogram is that the majority of these isopotential transcripts are representing exactly one gene, which is what we would have expected. And then you have several with 2, 3, 4, and so forth. Different transcripts, which kind of makes sense biological-wise, and this will be interesting to study. However, as you can see, we also have genes where we have 500 to almost 600, very few of them, but still potential transcripts overlapping each other, and we have to dig in here manually to decipher what is really going on. But this will be very interesting in the near future. So the ortholog comparison between our cavefish about these potential transcripts and the Mexican cavefish is currently still running. We are using OMA standalone tool developed by Christoph Bessimos in the University of Lausanne in Switzerland to dive into this interesting data set. So now that we have a nice reference genome that is actually phased, we have isoforms predicted and annotated based on these contexts. We of course want to dive in and look at the different mutations between the different caves and surface population fishes to understand better which alleles are common in one and not common in the other, as well as other population genetics calculation, evolutionary calculations. So to do that, we used pool sequencing. For those that are not familiar with pool sequencing, what is basically done is you take small aliquots of DNA from different individuals and mix them together in one sample and sequence this kind of mixed sample. So you see that on the right side, on the upper side of this plot, on the whole genome sequencing, where it's illustrated with flies. I took that picture out from a recent Nature review in genetics. And you can see what you get out of it is basically a mixture of different reads originating from different samples or different individuals of that population. Now the good side is that for the surface population, we were able to reach. For the pool sequencing, what is recommended is to have at least 40 to 50 individuals of the population pooled together to have a good representation of this population. For the surface population, this was easily achievable because the population on the surface is quite large and we could sample enough individuals. For the cave population, however, this was not possible because, again, as I said, this population is not huge and we didn't want to risk reducing the population size in the cave too dramatically to ensure the survival of this cave population. So overall, we just have like 34 different cave fishes pooled together in this Illumina sequencing run, which is not ideal, but hopefully is sufficient to estimate or get good estimations of these allele frequencies. So while we're doing this pool sequencing, well, it's a very cost-efficient way as well to get this population information, because instead of sequencing each of these individuals, we're just sequencing the pool. The other thing is not much DNA material is required per individual, so we didn't have to kill all these fishes and just use it for each of them, an Illumina, a part of an Illumina lane or a Illumina lane. It still provides the allele frequencies, and you can also estimate FST and other metrics that can be computed. The group from Vienna, from Schlötterer, published a nice population mathematics tools that can help you in the analysis if you have pool sequencing data. However, what we cannot assess with this pool sequencing data is, of course, phasing on its own, because we have this mixture of individuals and we cannot rely on which paired ends are coming from the same or from different individuals. So this is kind of a limitation of this otherwise very nicely method. So again, this analysis is still a little bit going on. What I can show you here is basically the allele ratio between the cave and the surface population on the x-axis, as well as the frequency on the y-axis. And you can see what we would expect is that since the split is so recent, as I said, 15,000 to 12,000 years ago, we see a lot of alleles that are being shared between the two populations, or not very different in allele frequencies between the two different populations, potentially rare alleles in the populations. But we see some on the right side that are very interesting, where we see a huge difference in the allele ratio between the surface and the cave population. So one of these examples that are pulled out is one potential gene here. So here I'm showing you on the right side an IGV screenshot with the isoseq data from the cave population in the top, where you see we aligned the different candidates to one of these regions. Then you see the pool sequencing Illumina data in the middle from the cave population. and you see the pool sequencing from the surface population in the lower track. And the reason I'm showing you exactly this picture is it kind of shows what we would like to see in some cases, that we see multiple mutations private to the surface population that we don't see in the cave population. Now, the reason why you see as few mutations here in the cave population is practically just that the de novo assembly that we produce with Falcon Unzipped is from the cave population and not from the surface population. So exactly this kind of genes and candidates of genes are very interesting right now. We are studying these candidates together with the ortholog information. As you can see in the iso-seq track, there might or might not be different splice variants. We're currently digging deeper into this. Based on iso-seq3, we are seeing at least 4 to 5 different isoforms here, but I think they can be better collapsed and then better explain the whole gene and what is going on in terms of isoform identifications. So what are we up to in kind of the next steps? Is we are currently working together with Phase Genomics to produce a high CMAP and do the scaffolding. So this is very interesting because Phase Genomics together with Barbatula developed Falcon Phase, which is a kind of a next step from Falcon Unzipped, which leverages this high C data to improve the phasing and improve this and apply the scaffolding in an automatic fashion. And we are very excited to see how this looks like. This is currently underway. The other thing that I'm super thrilled about and I want to share with you today is this little fella that I called Herbert, just of randomness. So Herbert is actually the first offspring of our cave population. So we were able to cross the different cave fishes that we currently have in our labs together and produce viable offsprings. And this is pretty neat because first of all, this allows us to have multiple follow-up functional studies on these cave fishes. And one thing that we're currently doing is growing Herbert as well as his siblings in part of this in a light condition and in part of a dark condition to really see if the conditions that are growing in influences some of the phenotypes that we observed in these cave fishes. Furthermore, we are currently generating F2 backcrosses between the cave and surface population to further study the genetics and genomics of these animals. So to kind of summarize what I've showed you today, and as I said, it's unfortunately still a little bit of work in progress, but it gets me very excited already. So we have already established, or we have already the first loach genome. It's a contig assembly. Very fascinating about this is about 70% phased contigs. So this is not just one sequence, it's really representing the diploid state of the genome itself of this cave population. We obtained iso-seq data to get an isoform, so transcriptome annotation of this genome. We are going to leverage, of course, computational annotation tools as well to complete the predictions of genes. We think we are already close to the estimated number of genes, but we are going to investigate this further. And then on the mutation side, we are currently analyzing the mutations that are different in the cave fishes as well as in the surface population and see which genes that are standard candidates for this cave fish evolution based on the investigations over the Mexican cavefish show different mutations. And I really want to stress this point because this is quite intriguing that the Mexican cavefish, and I maybe forgot to mention it, the Mexican cavefish is nowhere close related to our cavefish. So this means that if the same mutation is happening on the similar genes, then this is really independent evolution taking place just because of the selection pressure of the environment. But of course, we are going to look into that if that's the same genes or if these are then different genes. And thanks to the recent split, as I said, 15 to 20,000 years is current estimations. We are really excited to see how this rapid selection or this harsh selection pressure of this cave form these mutations in these genes itself compared to other cave fishes that have multiple millions of years been split from the surface itself. So to wrap things up a little bit, and as it was said in the introduction, that I'm more a computational guy than actually an evolutionary or biologist, I wanted to give some lessons learned that I saw, and as well as my collaboration partners saw, in analyzing these data sets and conducting this project. So first of all, as you might have thought, but still multiple people make it sound as easy as it is, de novo assembly and gene or isoform annotation is still not tricky. It's not a push button thing. You need to investigate a lot. You need to invest some time in it. Some of the challenges that I want to point out, it's still very hard to, almost impossible to really evaluate which of the assemblies, so in this case here we ran 3 different assembly tools, is really performing the best assembly. Some people are using N50, some people are using, for example, tools like Busco. Busco gives you core genes, non-duplicated core genes, and by that kind of an estimation how complete your genome is. But these are all rough estimates. This is not a kind of a clear solution, quite frankly, by any means. Another thing that I think is still lacking and would be nice to see some investigations in is how we can merge and leverage, or kind of a consensus approach, these different de novo assemblies together. You often see when you do different de novo assemblers on the same data set that are joining together different regions sometimes, or extending certain regions than other assemblers do. And it would be nice to dig in, or have a method to dig in and see what kind of support these connections have and how likely those are compared to different approaches. On the gene isoform annotation, it's even a little bit trickier, I would have to say. Just because the field is much younger, it's really hard to assess the quality of the data. As I said, most of the times the transcripts or the transcript to gene relationship is just 1-to-1 or 1-to-2, which kind of makes sense biologically speaking. However, we also obtain certain genes with potentially thousands of transcripts, which I would say is rather unlikely, but would be cool to see. And we are going to dig into that. And then it's also hard to assess or hard to estimate how many genes would you actually, or percentage of genes would you actually expect to be recurring from 1 or 2 flow cells. So this is the data that we currently having in sequel 1. It might change with sequel 2, of course, but these are kind of the questions that I think still needs to be answered. And the methods, I would say, are still very hard to control and understand where they draw certain thresholds. For example, from ISO-seq 2 to ISO-seq version 3, the pipeline changed quite heavily. And with that, also the estimations of potential transcriptomes changed quite heavily. And it's very hard to understand what is the benefit, what is the disadvantage of these 2 different versions. So with that, I want to bring it to an end. Acknowledge my postdoc Mahmoud at the Human Genome Sequencing Center, Richard Gibbs, my department head, who was very supportive also of this study, Arne Nolte, University of Oldenburg, as well as Jasminka Berman-Gol from the University of Constance. We 3 form these collaborations, and really many thanks to Jasminka who realized the importance of this fish and kind of drove this forward. PacBio for the smart grant, especially Sarah Kingen for the help with the optimization of Falcon and SIP, as well as GeneWiff for help with sequencing of the genome sequences, as well as from the ISO-seq libraries. Phase Genomics, with whom we are currently collaborating to bring this genome further to hopefully almost chromosome level scaffolds using Hi-C, and NIH for generous funding of myself. Thank you very much.
That was great, thank you. We're going to move to the Q&A now. We had some questions come in. First question, have you investigated the different orthologous genes between cave animals and how they relate to your cave species?
Yes, so this is an important question which is currently still underway. We are currently relying on OMA standalone, but there are different other methods. The interesting part is how these different genes look in terms of mutations between the Mexican cavefish. Unfortunately, I don't think there are many other cave fishes around that have been annotated in such a quality as the cavefish itself, but we pulled into the orthology study different other fishes that are not cave fishes, as well as certain outgroups such as zebrafish in humans and gorillas and chimps.
If the mutations and thus adaptations are occurring so rapidly, what does that mean for other cave organisms? Do they adopt rapidly as well and then specialize, or do they just keep constant over millions of years?
Right. I think this is like an important question that I'm not frankly able to answer myself. It's quite intriguing to see that the selection is happening and so rapidly in these fishes. So if you just go back to this picture, you can clearly see a distinguishment just between those two populations within the last 15 to 12 to 15,000 years. What this means for cave fishes such as Mexican is kind of an interesting question. If they have evolved very rapidly and then kind of specialized over time, or if this evolvement and adoption is happening over really constantly over these millions of years is going to be an interesting question, but we need much more data and much more cavefish studies or other cave animal studies, I guess, to answer that more clearly.
How important do you actually think phasing and isoform annotation are for these kinds of novel genome experiments?
Right. So in a standard project setting like this, you would be happy to just have a genome on its own, and then maybe either computational wise or with some Illumina sequencing, the gene prediction on its own. I think the current trend that we see, especially for model organisms, is that we are now taking huge efforts to improve our understanding of haplotypes and the role that haplotypes play in certain gene regulations and other regulatory units, and therefore also in phenotype between different humans and other model organisms. As well as huge efforts, I think from gene code, for example, to improve the transcriptome annotation from humans and other model organisms. So I think by looking at these kind of trends, it makes clear that we should start to approach these things and not neglect them. Also, when we're just talking about quotes, here about the first assessment of these cave animals. I think it's very intriguing and very important to try to bring in haplotypes and isoforms in this equation of evolutionary biology.
What follow-up experiments do you currently have planned to validate the impact of the mutations that you're detecting?
Right. As I showed, we are having this kind of crosses between the cave population itself that we're currently growing in different conditions, as well as these back crosses. This question can probably be better answered by my collaborators Anna Nolte and Jasminka, from University of Constance and Oldenburg. So I hope we can do at some stage also some CRISPR work on these animals to identify SNPs, but also potentially I would love to see some structural variants that we can show have some impact in gene duplications or gene losses between these different surface populations, or between orthologous genes of the Mexican and the cavefish population.
Last question, do you think that convergent traits between cavefish populations might have underlying genetic similarities in terms of mutated genes?
That is a tricky question, I would say. I don't know if I have an answer to that, because right now it's for me it's so the interesting part about this cavefish, it's our cavefish in Germany, it has the similar phenotypes to the cavefish in Mexico, similar, not the same, which if it's the same genes, it would mean that's independent evolution. If it's different genes, it's still very interesting, because then we have different mechanisms at play. But since I don't fully understand this role right now, I'm not fully capable, I think, of answering that question or have a thought about that.
Looks like in the interest of time, we'll have to stop here today. Once again, I'd like to thank Dr. Sedlazeck for his presentation and all the attendees for joining us. If you'd like to watch this webinar again, the recording will be available at genesweek.com. Also, check out genesweek.com for more activities, including webinars, exclusive promotions, multiple chances to win a total of $20,000 credit for Genomics services, and much more.